It's what's for dinner? I mean, it sounds simple, but for them, it's literally a life or death decision, and it all boils down to being super efficient with energy. Today, we're going to dive into the absolutely elegant genetic switch E. coli uses to manage its menu. It's a masterclass in survival called the Lac Operon.
You got to remember, for a single-celled organism, every single bit of energy counts. Making the right proteins and enzymes to digest a specific sugar, well, that's a huge investment. If you waste that energy making tools for food that isn't even there, you're going to lose the evolutionary race fast. So how on earth does it make the right choice every single time?
All right, so let's get specific. Let's talk about our little friend E. coli. It can chow down on a sugar called lactose, but to do that, it has to build some very special enzymes. And here's the dilemma. Building those enzymes costs a ton of energy.
Think about it, right? It's like you build this massive, expensive factory and then just let it sit there because no raw materials ever show up. What a waste. To solve this exact problem, E. coli evolved this brilliant little genetic switch to manage its production line. And that solution? It's the Lac Operon.
Okay, so the best way to wrap your head around the Lac Operon is to picture it as this super efficient, on-demand lactose factory that's built right into E. coli's DNA. It only fires up the assembly line when the main ingredient, lactose, is actually on the scene.
So let's get a bit more technical for a second. The Lac Operon is basically a stretch of DNA that holds the blueprints for those lactose-digesting enzymes. And the key word here, you're going to want to remember this, is inducible. It means its activity is induced or triggered by the very molecule it's designed to destroy. Pretty cool, huh?
All right, to really get how this factory works, you've got to meet the key players. First, you've got the structural genes. Think of these as the actual blueprints for the enzyme machinery. Then you've got the promoter. That's kind of like the docking site, the big start here sign for the enzyme that reads the genes, RNA polymerase. Next is the operator. This is the all-important on-off switch. And finally, we have the repressor protein. This is our security guard, and its whole job is to block that switch and keep the factory shut down. And here you can see just how perfectly everything is lined up on the DNA. You've got the promoter, which is the entry point, the operator, which is the gate right after it, and then the genes, which are the factory floor. This super precise arrangement is exactly what makes that on-off switch so incredibly effective.
Okay, so what's the factory's default setting? When there's no lactose floating around. Well, the whole system is designed to be completely off. It's all about saving energy.
And here's exactly how it stays shut down. That repressor protein, our security guard, literally sits right on top of the operator switch. It acts as a physical roadblock, making it impossible for the RNA polymerase to slide down the DNA and read the genes. Simple as that. No lactose, no enzymes. The factory is closed for business.
But what happens when some lactose finally shows up on the menu? This is where the design gets really clever. The factory needs to power on and fast.
When lactose gets inside the cell, it acts like a key. It finds that repressor protein and binds to it, which changes the repressor shape and makes it fall right off the operator switch. The security guard is gone, the roadblock is cleared. And bam, RNA polymerase can now lock onto the promoter and start zipping down the line, transcribing those genes into enzymes. The factory is officially open.
But hold on, it's not quite that simple. Just when you think you've got it figured out, there's a fascinating little twist to this story. Turns out, E. coli is actually a bit of a picky eater. It has a favorite food, and it will always choose that one first.
And that favorite food is glucose. It's just easier to break down. You get more bang for your buck energy-wise. So E. coli has a second layer of control built in. It's not going to bother running the lactose factory at full tilt if the much better option, glucose, is right there for the taking.
So check this out. When glucose is present, the repressor might be off the switch, but the RNA polymerase, the machine that reads the genes, just can't seem to get a good grip on the promoter. It's really unstable. It's like it's trying to work on a slippery surface. It just keeps falling off. So only a tiny bit of transcription happens. The factory is technically open, but production is running at a snail's pace.
Now, the factory only kicks into high gear, we're talking full power mode, when the good stuff, glucose, is totally gone. When that happens, this little hero molecule called an activator protein swoops in. It grabs onto the DNA and acts like a clamp, holding the RNA polymerase firmly in place so it can do its job properly. This is what cranks transcription all the way up to maximum speed.
So why does all this matter? Well, the really crucial point here is that this isn't just a simple on-off switch. It's more like a sophisticated little computer, a logic gate that takes in information about two different sugars to make the absolute best, most energy-efficient decision possible.
Okay, this table really lays it all out perfectly. Let's just walk through it. Look at these 4 different scenarios. Glucose and lactose present? Nope. Just glucose? Definitely not. Neither one? Still nothing. But notice something? There is only one condition, just one, where we get real transcription, and that's at the bottom, when glucose is absent and lactose is present. In every single other case, the factory is either completely blocked or running too slowly to even matter.
And this is the big takeaway, really. This incredibly elegant system in a humble little bacterium ended up becoming the foundational model for how we understand gene regulation. It was the first real proof that genes aren't just static blueprints. No, they're dynamic, responsive systems that can be turned on and off by signals from the environment. And this basic principle applies to all life, including you and me.
And that just leaves us with this one kind of mind-blowing thought. The Lac Operon is just one of thousands upon thousands of these genetic programs. Your own cells, right now, as you're watching this, are running countless, tiny, sophisticated switches responding to everything from the food you just ate to the light in the room. So, what incredible genetic programs are running you?